Dear students, I am Dr. Nidhi Sharma, p.g.govt.college, Jhalawar, Associate Professor, Zoology. Students, in continuation of my earlier video of genetic recombination, I am telling you something about conjugation in this part. Conjugation requires direct contact between cells. Only It is the only form of gene exchange in which donor survives. It is mediated by plasmids. The plasmids can can code for traits that give bacteria advantage. Plasmid types are fertility factor, dissimilation plasmids, resistance factor plasmids, bacteriocin factor plasmids, and virulence plasmids. Now, how does combination takes place? Here we have a bacteria which is F positive, which has a factor positive cell, and another bacteria of the opposite strain having factor negative cell. Now, this has an F plasmid and this does not have an F plasmid. Now, the second stage, in the first case, the donor cell, which is the F positive cell, attaches to a recipient cell with its pilus. You can see the conjugation pilus in this diagram. The pilus draws the cells together. In the second stage, the cells contact one another. In the third stage, one strand of plasmid DNA transfers to the recipient cell. In the fourth stage, the recipient synthesizes a complementary strand to become a F positive cell. The donor synthesizes a complementary strand, restoring its complete plasmid. Here you can see the donor chromosome now, which has become the F positive cell. This donor chromosome, which is the F positive cell, the plasmid integrates into the HFR cell. Now, the HFR cell again forms, which is now F positive cell, HFR cell. Again, applies is joined, pilus is joined and with the F negative recipient. Here, in the first stage, F plasmid integrates into chromosome by recombination. In the second stage, cells join via a conjugation pilus. In the third stage, portion of F plasmid partially moves into recipient cell trailing a strand of donor's DNA. In the fourth stage, conjugation ends with pieces of F plasmid and donor DNA in recipient cell. Cells synthesize complementary DNA strands. Here you can see incomplete F plasmid cells remain F negative. Now we have got the recombinant cell which is F negative. In the fifth stage, donor DNA and recipient DNA recombine, making a recombinant F negative cell. Here you can see the elaborated diagram of the base pairs. This is the electron in the A instance electron micrograph picture of this plasmid. You can see you can see the origin of replicon. There's a MER gene, SUL gene, STR gene, CML gene. These are R determinants. TET gene's origin of transfer starts from here. RTF and pilus and conjugation proteins are present, and that is how this gene is a recombinant gene. Thank you.